Welcome to Science Café. This is the third of our summer virtual Science Café series. I suppose you could say we didn't have a choice with either it being summer or having to go virtual. But welcome to this physically distanced cafe. I'm Pam Wolf. I'm on the chemistry department faculty at Carleton University, and I've been hosting Science Café for about the last 5 years. Usually in person in the Sunnyside branch of the Ottawa Library, and now here on Zoom over the summer. Science Café itself's been running since 2008, so 12 years now that we've been putting these cafes on. And they were designed to give those of the public who were interested the chance to find out the work that's going on in the science department at Carleton University, and to give Carleton University researchers the chance to talk to the public who support them about the work that they do. So we definitely see it as a win-win situation. We also take quite seriously our responsibility to tell people about both fundamental and applied research that's going on, because for people who don't have a day-to-day connection with the science research community, or a research community of any sort, the value of that fundamental research isn't always immediately obvious, and it's very easy to fall into the trap of looking at the surface of something and thinking, that sounds like a remarkable waste of time and money. And if we're not out talking to people about how very, very important that all of that work is, we really can't complain about that. So the Science Cafes were designed from our point of view to allow us to talk to people about the things we're passionate about and the things that we feel are very, very important. And I think there's no time like the present to make it so clear how having a fundamental background already in place has allowed researchers in Canada and around the world to hit the ground running, doing applied research that 6 months ago they might not have pictured themselves doing, but 6 months, 6 years, in some cases 16 and 26 years ago, they were laying the groundwork for by studying fundamental things. So that's the whole roundup of Science Café over the last 12 years, and why we feel it's particularly relevant to keep meeting like this, even if we're only meeting online rather than together. Today's speaker is an excellent example of everything that I've been blathering on about. Dr. Alex Wong is a professor in the biology department at Carleton University. He's been at Carleton since 2013. And at Carleton, he does both fundamental and applied research in the field of experimental and computational methods, studying evolutionary biology, primarily on bacteria. He did his PhD in genetics at Cornell, and then did postdoctoral fellowships in the Ottawa area at both Carleton and the University of Ottawa. And among other things, up till a few months ago, one of the focuses of his research and his research group was antimicrobial resistance. With the COVID-19 pandemic, he's been collaborating with local hospitals and private companies to study the decontamination process of personal protective equipment, primarily for people in the first responder and healthcare fields, although of course these are going to be important for all of us moving forward. To enrich his working from home experience, he has a wife, 3 children, and a dog. And he runs and plays the bass guitar in whatever spare time he used to dream about having before the crisis hit. So I will now turn over to Alex to share his screen and his work with all of us.
Great, thank you, Pam. And thank you everybody for dialing in this afternoon. I really appreciate you spending the next little while with me.
And what I didn't put in my notes, but I want to repeat, before I let Alex start, he can get his slides going, is that we will be taking questions. If all of the participants look down, you have a question and answer box. As you think of questions, for goodness sake, please do type them in. We will track them, but we are going to answer them all at the end because this isn't quite as interactive as an in-person. So please do ask them as you think of them, but we'll deal with them and get to them at the end. Sorry, Alex. Now it's yours.
That's okay. Thanks a lot, Pam. And again, thank you everyone for being here. I appreciate it. So as Pam said, my name is Alex Wong. I'm an associate professor of biology at Carleton. Under normal circumstances, my lab does work primarily on the genetics and evolution of bacteria. We do a lot of work on antibiotic resistance. However, in the last few months, we've been turning our attention to COVID-19 and trying to help out where we can. Today, I'll be telling you about some of the work we've done on, and that we will be doing on decontaminating medical masks using ultraviolet light. So we are here, of course, because we're in the middle of a global pandemic caused by the SARS-CoV-2 virus. So SARS-CoV-2 is the name of the virus, and the disease that it causes is COVID-19. As we know, COVID-19 first emerged at the end of 2019 in Wuhan province, and quickly achieved global distribution early this year. In Ottawa, we've seen just over 2,100 cases as of a couple of days ago, and about 260 deaths. Fortunately, we've done a pretty good job of sort of flattening the curve recently, and we're only seeing a handful of new cases every day. What's really emerged both in Ottawa and worldwide is that healthcare workers and first responders are particularly at risk for infection with COVID-19. And that's probably not surprising. We know that the virus is spread when sick people cough or sneeze, and they release droplets that are packed with virus, and then someone breathes in those droplets and gets sick. First responders and healthcare workers are, by the nature of their jobs, going to be exposed to more sick people, more people coughing, more people sneezing, and so they'll just get exposed to more virus particles and are going to be more likely to get sick. And so in Ottawa, something like a quarter of the cases that have been documented have been in healthcare workers or in first responders. And I should say that that case count in Ottawa is probably an underestimate. Those are confirmed cases, so people that have gone in and had the laboratory test done. There are probably many more people who have gotten sick, but who haven't had the tests done, and so we can't count them in the case total. So the best defense that healthcare workers and really everybody have against SARS-CoV-2 is prevention. As the old saying goes, an ounce of prevention is worth a pound of cure, especially when you don't have a cure, as is currently the case. So the sort of standard protection prevention equipment for a healthcare worker is shown here. So this is the personal protective equipment, or PPE, that someone working in a hospital might wear. So they'll have disposable gloves that they can change between patients or between duties. A gown and a hair cover that will protect against bringing the virus home on their clothes or their hair, goggles to avoid exposure to the eyes, and an N95 respirator that will block exposure through inhalation. Now, you probably remember that early on during the pandemic, there were really severe supply chain disruptions for N95 masks. We found ourselves in a situation where the supply of N95 masks that we had in Canada was insufficient for the number of cases we were worried would emerge. So if we started seeing hospitals swamped with COVID-19 cases, we didn't think we had enough N95 masks to go around and to protect the healthcare workers who were on the front lines. Thankfully, over the last couple of weeks, it seems like we've really remedied those supply chain situations. So there's been a lot of hard work put into sourcing masks from different suppliers. A lot of local companies and Canadian companies have, that were manufacturing other products have re-geared their production lines to produce N95 masks. So it sounds like we have a good supply of N95 masks now. But what this the supply chain issues really did was highlight the importance of having a safe and consistent supply of PPE. It really quickly became clear that we weren't prepared and that we needed to have some safeguards and checks in place for our PPE supply. And we'll come back to some of these issues in a little bit. So the N95 mask is, again, we've heard a lot about this over the last few months. Many of us probably hadn't thought about them before, or we only thought about them if we needed them for some home rental project. But really, N95 masks are a mainstay of safety for people, not only in industry, but obviously in the hospital. So, when I got into this, I started wondering what the N and the 95 actually refer to. So it turns out that the 95 part of the N95 mask means that this is a mask that will filter 95% of particles out of the air. And we're talking very, very small particles, so particles that are 0.3 microns in diameter. So one of these masks, again, will filter 95% of those particles out. The N part of the description refers to how effective a mask is in the presence of oil. So in certain, let's say, industrial applications, you might have an environment where there's oil particles in the air too. An N95 mask is not effective in the presence of oil, whereas other kinds of masks are. But in a healthcare system or a situation, you wouldn't expect there to be a lot of oil in the air. And so an N95 mask is very good protection. The history behind the N95 mask is actually pretty interesting. It's worth following up on if you have the time and inclination. The filtering technologies, so the way the mask material itself is manufactured, was developed by Peter Tsai. He's a material scientist at the University of Tennessee. Dr. Tsai, I guess, retired not too long ago and recently came out of retirement to help study mask decontamination techniques. The shape of the mask and the sort of manufacturing process were developed by Sarah Turnbull. So Sarah Turnbull is, I gather, quite a famous designer, American designer. She passed away a few years ago. And for a while, she was working at 3M, which is one of the big manufacturers of N95 masks now. And she had been working on a number of design projects, one of which was the design and manufacturing of women's undergarments. And she had the idea of applying the technologies that are used to shape molded bra cups to mask formation. And so those techniques are still used to produce N95 masks today. So coming back to the issue of decontamination, when we were worried about supply chains and these supply chain shortages, decontamination emerged as a method to sort of prolong our supplies of N95 masks. So normally in a hospital, a doctor would, or a nurse would, discard their mask after each use to avoid making other people sick with whatever's on that mask. So the idea here is you have some method that allows you to decontaminate the mask, and you can use it repeatedly. And so this would stretch out your limited supply of N95 masks. So as I've talked about, Canadian supplies are pretty good right now, but you can imagine a number of situations in which decontamination can still be useful. So if you're working in a remote setting, so let's say Northern Canada, maybe it's hard to get masks up there quickly. So having some method for decontaminating the masks that you have on hand would be really useful. Similarly, in mobile operations, let's say with the military or the Red Cross who are going out mobilely to remote locations, having a method for decontamination would be useful. Decontamination is likely to be useful internationally as well. While we seem to have COVID-19 currently under pretty good control in most of Canada, the international situation is still not so good. So what I'm showing you here is the growth of COVID-19 cases worldwide over the last several months, currently at 11.5 million, and that's still a steeply increasing curve. And so there's potentially still need for decontaminating procedures around the world. So there are a number of methods that have been looked at before that seem to be pretty good for decontamination, depending on the situation you're in. So for example, hydrogen peroxide is a probably a good decontamination method, at least in sort of larger hospitals and larger healthcare centers. So hydrogen peroxide, this is the same stuff that you would go into the pharmacy to buy and use to disinfect a cut. This is the molecule here. So when in a decontamination context, the hydrogen peroxide gets volatilized, it gets released into the air, and you're in the masks are placed in a sealed chamber with this volatilized hydrogen peroxide, and the hydrogen peroxide kills whatever's on those masks. It's very effective and potentially large scale if you have a large enough chamber. The equipment that you need to do this is quite specialized, and so it's probably only a viable solution for large permanent locations, things like hospitals in big cities. So it'd be good in Ottawa or Toronto, maybe not in Northern Ontario. Heat is also a good decontamination method. So if you heat the masks up to 60 or 65 degrees Celsius for a prolonged period of time, you can kill most of what will be on there. The one problem with heat is that it will deform certain masks. So some masks have like a plastic piece that forms the shape around your nose, and that can get deformed by the heat treatment. The method that we've been looking at is irradiation with ultraviolet C, and I'll talk some more about that. So what I'm showing you here is a cartoon of the spectrum of different types of radiation. So different kinds of radiation, so visible light and infrared and ultraviolet light, differ in their wavelengths. So the visible light that we see every day is roughly between 400 and 800 nanometers. And just below that, between 100 and 400 nanometers, are different kinds of ultraviolet light. And we typically divide up ultraviolet light into three categories: long wave, middle wave, and short wave. Short wave is the set of wavelengths that tend to get used for germicidal applications, so decontamination of masks would fall in there. And those are wavelengths between about 180 and 280 nanometers. We got into this project through some contacts at the Children's Hospital here in town. So early on during the pandemic, CHIO got really interested in mask decontamination methods because of the anticipated shortages. They conducted a number of really nice meta-analyses and systematic reviews looking at literature that had already been published on using different methods, to decontaminate masks. So these wouldn't necessarily have been studies that had been carried out on SARS-CoV-2, maybe on flu, or maybe on other surrogates of infectious diseases. But the methods that work for, let's say, flu probably work for SARS-CoV-2 as well. So the CHIO group has published a number of papers on these topics. This is the one most relevant here, where they reviewed a lot of literature on ultraviolet decontamination. Now, I won't go through everything that they found, but the take-home message was that, yes, indeed, ultraviolet C irradiation does seem to be an effective decontaminant. So these two plots here show that when you increase the UV treatment, going to the right on the plot, you get a stronger and stronger reduction in viral load. So you're killing the virus. There are a number of questions that are still open. So these data come from, depending on the question, about 6 to 12 studies. And one of the ones that we're interested in looking at is the question of wavelength. So the most commonly used wavelength for decontamination purposes is 254 nanometers. So going back to this plot, we're in the short wave spectrum. So our 254 is within that spectrum. And all of the studies that were reviewed in this paper use that wavelength. So we've been interested, among other things, in whether other wavelengths are also practical for UV irradiation. And one of the motivations for doing so is the availability of bulbs and LEDs that produce those wavelengths. So LEDs at longer wavelengths, so closer to 280, sort of 270, 280, get used in other applications, like in horticulture. So those light sources are just more available and cheaper. And so if people want to start manufacturing these things on a larger scale, there might be some value in using different longer wavelengths. So we've been working with a local company, Arcalumen Incorporated, on asking these questions. So Arcalumen is a local company that produce LED lighting solutions. They make custom lighting modules and LED controllers. And when the pandemic hit, they started to think about, well, how could they help, and what are some of the kinds of products that they can envision that would help with some of these issues. And so working with CHIO and with Arcalumen, we're looking at, again, the effectiveness of some of this UV decontamination. So Arcalumen provided us with the unit I'm showing you here. This is a custom prototype cabinet with LED lights. So I hope you can see my mouse. So at the back of the cabinet here, these boards have 4 sets of LED lights. So these little circles you can see in the middle are the LEDs. When we close up the cabinet and turn it on, they emit in the sort of 275 to 280 nanometer range. There's hooks on the doors so that you could just hang your N95 masks on them. And I'll show you what we've got here in a second. And then this is just a monitor that shows us how much UV light is actually being emitted. So we don't want to sort of plow through hundreds and hundreds of masks to do our tests. So for the time being, we're just using sections of masks. So we cut up N95 masks into pieces, and we inoculate organisms onto those to do our tests. And so we mount these little pieces of masks on cardboard inside the door. That's what these are. And then we can test the effects of different amounts of UV irradiation on the load of organisms on those pieces of mask. So we're just at the early stages of this research. We haven't yet been doing any work with SARS-CoV-2. Ultimately, so we won't be using live SARS-CoV-2, but we can do a lot of experiments actually using inactivated SARS-CoV-2. We haven't been doing those experiments yet. Our preliminary experiments have been carried out using a bacterium, E. coli. So E. coli is a resident of our guts. It sometimes causes disease, but not usually. We use a strain in a lab that is unlikely to make you sick. It's easy to work with, it's safe. And so it's a good organism for getting a handle on the system, figuring out how to use it, figuring out roughly what sort of range of doses we're going to be looking at. So our experiments look something like this, and I'm going to show off my poor skills at making art in PowerPoint here. So we start off with a tube of bacteria. We just grow up some E. coli overnight. And then we apply it to a mask, or in this case, our pieces of mask that we have inside the chamber. So this is a piece of mask that we've applied some bacteria to. And again, that's these little squares that we mount on the inside of the cabinet. We then irradiate those pieces of mask for different amounts of time. So we will either not irradiate them at all, or irradiate for 10 or 20 minutes or 30 minutes, depending on how much of a dose we want to administer. And finally, we can count bacteria. And by comparing the undosed bacteria versus different times, we can get an idea of how effective that treatment is. So here's an example of the kind of data we generate. So what I'm showing you here on the x-axis on the bottom is the dose of UV irradiation. The units here are in joules per meter squared. This corresponds roughly to time. So the highest doses are after about 40 minutes of UV exposure. On the y-axis here is the number of bacteria that we're able to recover from one of those squares of mask. This is on a log scale. So this is 10 bacteria, 100, 1,000, up to 10 million, or a million is what we actually see. And so what you can see is that when we don't dose the fragments of mask at all, we have a quite high load. We have about 1 million bacteria per square. As that dose increases, at the highest exposures, we have essentially no bacteria that we can recover. So this shows us gratifyingly that we can kill an organism using this UV exposure, which we would have expected. Importantly, this is really repeatable. So each of these data points is a separate replicate, and each of the colors is an experiment we did on a different day. So we can get consistent results on the same day or between days. So importantly, this is a repeatable result. And it's pretty quick. We can get pretty much complete killing after about 30 minutes under these conditions. And there are things we can envision doing that would make that faster if we increase the power to the LEDs, for example. So we still have lots of work left to do. As I said, this is really just preliminary work that we've done in E. coli. And what we really want to get at, of course, is effectiveness against SARS-CoV-2. And these are the experiments that we'll be doing over the next few months. So hopefully we'll have more to share on that by the end of the summer and early fall. And with that, I'd like to thank all the people who have been involved in this work. Aaron Hines is a research associate in my lab. He's been with us for about 3 years, and he's done all of the work that I've showed you. So huge thanks go to Aaron. I want to thank our collaborators at CHIO, in particular Richard Webster, who was really instrumental in getting us involved in this work. Of course, our industrial partner, Arca Lumen, who have been also excellent partners on this work. They've provided the prototype UV cabinet and a lot of feedback and interaction on the experimental design. We've gotten some funding together with CHIO and Arca Lumen to do this work from NSERC, as well as from Carleton. And of course, I'd like to thank you. I really appreciate your being here today, and I'm happy to take questions. Thank you.
Okay, thank you very much, Alex. We do have some questions in the Q&A already.
Okay.
And the first one that came in was presumably what you were saying was the existing technology that were in the meta-analyses.
Mm-hmm.
The question was, why 254 nanometers and why not higher energy than that?
Yeah, so I think there's a fairly complicated set of answers to those questions. Part of it is it's not just a matter of energy. So different wavelengths seem to have different effects. And in some of the early studies, 254 seemed to give you sort of the most bang for the buck. It's targeting, probably DNA a lot of the time, and that particular wavelength of light seems to be particularly damaging for DNA. It's also a matter of availability and cost and that sort of thing. But 254, so for a combination of these reasons, 254 has emerged as the standard.
Okay, and how effective is the UV radiation which is being used inside airlines?
That's a good question. Yeah, I've seen some of these pictures of trolleys being pushed down the aisle of airplanes, apparently irradiating with UV. Okay, so first I'll say I haven't seen data from them, so I'm not, I'll be a little bit careful in my response. I am a little skeptical that there's going to be a whole lot of effectiveness there. So if you look at the times, so I mentioned a little bit about the time scale in which we're seeing killing in our experimental system. We're seeing that we need to expose our fragments of mask to pretty strong UV light sources for about half an hour to get appreciable killing. I don't think that's what they're doing on airlines. Maybe I'm wrong about that, but my impression is that that's not what they're doing. So I would like to see the data. I would like to see evidence that this is working. Until I see that, I'm a little skeptical.
And is the temperature at which the irradiation takes place, does that contribute to the effectiveness?
Yeah, that's a good question. I don't have a real good answer for that question. So I So I can imagine a couple of ways in which temperature might matter. So it might matter if if UV killing is sort of metabolism dependent. So, do the bacteria need to be actively dividing in order for the UV to be effective? I don't think that's the case. In the conditions that we're doing our experiment in, the bacteria are probably just hanging out and not actively they're not metabolically active. And especially for the viral stuff, viruses, SARS-CoV-2 on a mask is just going to be sitting there waiting to infect something. I could see an interaction perhaps at higher temperatures. So we talked at the beginning about how temperature can be used for decontamination too. So it might be the case that at higher temperatures there's some sort of interaction where heat plus UV maybe has a synergistic effect. I don't know that that's the case or not.
And I think as a chemist, the next question ties into that a little bit,
Yeah,
which
absolutely.
is the much more broad, how does UV light kill viruses and bacteria?
Yeah, absolutely. So the short answer to that question is it causes damage to a lot of different macromolecules. So it causes damage to proteins and to lipids and to DNA. A lot of at least some of the focus has been on damage to DNA and in the case of SARS-CoV-2 to RNA. We know quite a bit about the kinds of damage that are inflicted on DNA and RNA by UV light. And so we think it's just an accumulation of damage as you expose more and more to the UV, you're getting more and more damage.
And a very relevant, timely one. This week, there's a lot of talk about SARS-CoV-2 in the air. Could UV lights be used to eliminate airborne virus?
Yeah, that's a good question. And it's something we've been wondering a bit about too. So you might imagine a situation, maybe, I don't know, everybody goes home from the office at night and you have some low level of UV light that you shine on the office all night, or in your air circulators or something like that. And I think there might actually be some promise there. And especially with these aerosols that people are talking about. So I'm just going to give a little bit of background on Martha's question. We've been hearing for weeks now that most of the transmission is through the larger droplets that we cough out and sneeze out. Those droplets can contain many, many, many virus particles. There's a suggestion earlier this week that maybe much smaller aerosol particles might also be responsible for some transmission events. And the nice thing, well, it's not nice, but one for us benefit, if that is true, is that these smaller droplets are probably going to be more susceptible to UV irradiation because they're a smaller volume, there's a smaller number of virus particles, the killing should be much faster. So that was my long answer. The short answer is, yeah, there's probably some promise in using UV decontamination of air. I think there'd be some figuring out of the mechanism of how to actually do that, but I think there's some promise.
I have heard about installing UV in HVAC systems, and that is not new to COVID-19.
Right.
That has been discussed for numerous things that are known to be transmitted. I do not remember the details, but certainly I've heard of that. So I'm sure there are people working on ramping that up for the SARS-CoV-2.
Yeah.
Yeah. Yeah. Would plain old sunlight work, or is its UV too long?
Yeah, good question. So its UV is probably too long is the short answer. So the ozone layer blocks out the vast majority of the UVC that's present in sunlight, and so very little UVC makes it to the ground from sunlight. Now, it could be the case that we don't really know much. Well, one of the things we may end up investigating is how effective UVB is, so some of the longer wavelengths. We do know that there are some antibacterial effects of UVB, so it's an interesting question as to whether there are UVB effects on SARS-CoV-2.
Is the efficacy of decontamination dependent on humidity? And did you dry the pieces of mask before the irradiation of UVC in your radiation chamber?
Yeah. So again, I don't know if humidity matters. It again, one might imagine that there are ways in which it could. We haven't been drying the piece of masks for our experiment. That's probably something we should investigate. But yeah, no, we haven't looked at that yet.
Are there any UV lights that can be found at big box stores? So
Yeah,
is this a do-it-yourself at home
yeah.
technique?
So yeah, it's funny. So I Googled this a few days ago, and you can find all sorts of products, little wands and flashlights and stuff that are purportedly UV sources. Again, I think this is an area where we are best to be a little bit skeptical. So part of my reason for saying that is UVC can, it's great decontaminant. It's also can be quite harmful to people. So if you expose yourself to a UVC light source for not very long, you'll get a sunburn or eye damage. And so I don't think that manufacturers are going to be able to sell UVC lights that are very strong. And so you're going to have to get any appreciable decontamination, you would have to have a really, really, really long exposure from your UVC wand or flashlight. So yes, there are products out there that are advertised. I would not depend on those for decontaminating my my anything.
Have you thought of using animal coronavirus, swine avian, before going to the actual SARS-CoV-2?
Yeah, so we had to think about this quite a bit in designing our experiments. SARS-CoV-2 live virus is considered a level 3 pathogen, which is the second highest categorization. We're not certified to work on that. This is pretty high level stuff. I don't really want to work on level 3 pathogens. So we've sort of hit upon two alternatives to using live SARS-CoV-2. So one of them is exactly what you say. There are coronaviruses from pigs and mice and all sorts of things that you can buy that are level 2, and we are level 2 certified. The other option, which I briefly mentioned, is you can also buy SARS-CoV-2 that's been heat inactivated. So the virus particles are intact, but they're no longer infectious because they've been heat treated for whatever appropriate amount of time. We're gonna try to do our experiments with the heat treated SARS-CoV-2 more for technical reasons. Our detection of those viruses is using a molecular method called PCR, and we'd rather, if we use the heat-killed SARS-CoV-2, we can use the same protocols that medical clinical labs use. We wouldn't have to develop a new one for, let's say, a mouse coronavirus. But yeah, good question. Thank you.
And next one, bacterial genomes are much larger than the SARS-CoV-2.
Yeah.
Why not looking at other coronaviruses immediately as surrogates?
Yeah. So, I mean, I think we partially just answered that question. We are going to be looking at either heat-killed or other coronaviruses as surrogates. Really, going to bacteria first was a matter of what can we do easily and quickly to validate our methods while we're waiting for qPCR primers to come in, while we're waiting for viruses to come in. It was a matter of what can we get started with quickly. The UVC penetrations, yeah, that's a great question. That's ultimately one of the things we'll look at. So for the decontaminations that the hospital is doing now, at least the last I heard, they would expose one side of the mask, then flip them over and do the other side of the mask, under the assumption that the UVC is really not penetrating that deep. And that's a good question that we'll want to ask, especially since the don't know if you've ever taken apart an N95 mask, but they're in layers. And so there could be well be an issue of, if you only treat one side, you might not get contaminants on the other one. Good question.
And the question again relating back to something that you've addressed, but how effective is the UV cell phone disinfectant on the market right now?
Yeah, so again, so I don't know that particular product, but for the reasons I talked about earlier, I would be skeptical. For your cell phone in particular, it's easy to, what I do when I get home from the grocery store, I spray it down with hand sanitizer or wipe it down with some ethanol. And that's just as easy and probably more effective.
And this one too, I believe you've addressed. What are the major technical caveats with using the UVC short wavelength decontamination methods, penetration into layers of the
Yes.
N95? So yes, I think we've definitely talked about that one. And yes, you've also talked about sunshine, but I'll repeat that because there was a specific part of this question, which is the sunshine effective in disinfecting masks? And can we leave masks in the masks in the sunshine? She sells seashells by the seashore. Okay, I can do this. Masks out in the sunshine for a few days to inactivate the virus.
Yeah, so yeah, as we talked about before, I don't think the ultraviolet in the sunshine is necessarily going to do a lot to inactivate SARS-CoV-2. But that said, and actually the next question gets at this too, on most surfaces, on any of the surfaces that people have looked at, SARS-CoV-2 doesn't seem to last more than a couple of days. And so when you leave your mask out in the sunshine for a few days, really, I think it's time that's buying you decontamination and not the UV. If you want to go faster, you can, it's fine to just run your mask through the wash too, right? Soap and water are a plenty good decontaminant. Good question.
And this one certainly touches on exactly the same thing. Can I cycle through a different mask, say, every 3 days, as they say that COVID-19 doesn't survive more than 3 days?
Yeah, exactly. So that's same answer there. Yeah.
And next one, disinfection often depends on the organic load. How will you mimic the organic load that might be found on virus-containing droplets?
Yeah, interesting question. So, okay, so I hope I'm interpreting this question correctly. So by organic load, I think, Warren, you might be referring to non-virus material, all the other stuff in your saliva that's not virus. And maybe it's absorbing some UV light, providing protection to the virus particles. So we haven't gotten this far yet, given that we haven't started working the virus. But there are a number of ways you can imagine doing that. A lot of what's in your saliva will be dead skin cells and other bacteria. So you could imagine making a sort of a slurry of human cells and bacteria and looking at the effectiveness on decontamination. We have another SARS-CoV-2 project on the go, which is more about detecting SARS-CoV-2 on doorways and that sort of thing. And there we are looking specifically at, if there are other bacteria around, does it affect our ability to detect the virus? It's a similar sort of question. But yeah, no, thank you.
Okay, and looking to extend this decontamination, would these lower wavelengths be damaging to electronics such as phones?
Good question. I have no idea. Simply because I don't know enough about the electronic side of it. It's a possibility, but I'm not a good enough engineer to answer that question. Sorry, Remy.
They're certainly not going to do anything for the plastics and cases.
Right, absolutely, yeah.
But the actual electronics, I have no idea either. And next question. For those without access to UV equipment and N95 masks, can you advise what's the effective lifespan of the standard surgical mask, and are they that are readily available? And is this total lifespan extended if the usage is broken up by periods of non-use? And how long would that period of non-use need to be? Can these masks be cleaned?
I don't have a good answer to any of those questions, and I hesitate to come down too firmly on any of those questions, simply because I don't want to say something that will lead to people getting sick. I mean, those surgical masks, you know, I have a pack of them in the car, are meant to be single use. I don't know what, you know, the typical lifespan of a use is, or what the lifespan is. And I'm hesitant to say more than that. Thank you for your question, Gregory. I'm gonna have to apologize for for not giving you an answer.
And I'd like to sneak one in a little bit from the engineering side. You were mentioning the choice of wavelength being based to some extent on availability. What light sources are you using? Like the 254, I believe that you mentioned, and you said 272 now that you're working on. I've probably forgotten the actual numbers.
So the ones that we're using are LEDs, and that's a little bit different from what's used in other industrial applications. So often it's these mercury vapor lamps that get used for the 254. We're using LEDs because that's what our industrial collaborator are interested in. And there's I mean, on a broader scale, if we can use LEDs, there's a potentially lots of LEDs out there, and an ability to scale up that kind of an application with LEDs, which isn't necessarily there for other kinds of light source.
Okay, and I believe we've covered, oh, nope, my mistake. I need to scroll down. Like heat, is it possible that UVC could also compromise the structure of the mask or other PPE?
Yeah, so there has been some work done on this question. So I referred to the systematic review that CHIO did, where they looked at effectiveness of UVC. Some of the studies in that were reviewed in there also looked at effects on mask fit and air filtering ability. And there, the UVC exposure didn't seem to have an effect on either fit or sort of permeability. It's only been a handful of studies, and my guess is that there's probably some more that needs to be done there. You know, how much energy, as Pam alluded to, there's going to be some damage to plastics and that sort of thing. So I don't think you can sort of expose the masks repeatedly over and over and over again and expect not to see damage. There's got to be, I would imagine there's some limit. We just don't know where that is.
The first thing that came to mind for me is I suspect they would be changing the elastics on the masks rather than using them and risking having them snap, because the ultraviolet is going to have quite a profound effect on that part of it.
I agree.
But even in a mobile situation, that should be a fairly easy thing to do. Probably not a regular stapler, but when I look at my N95 masks, which I had from, I'm a stone carver, they're a specialized staple, but they're simply the attachments are stapled in.
Yeah, exactly.
And they're the first thing to go on the mask all the time. And I used to re-staple my elastics on.
Absolutely.
Yeah. The ARA has done trials with sebum mimicking oily skin and mucin, saliva-like, as vehicles. It seems to operate well in these real-world settings. Does your lab plan to explore this?
Now we do. No, that's a great point, Liam. And Liam and I have actually exchanged some emails before this.
Oh, so he's putting you on the spot in
He is
public
putting me on the
now.
spot. That's okay.
Well,
No,
of course it is. I was praising him, not criticizing.
This gets back to the question about organic load and what would be the appropriate uh, organic load to use, and those sounds like some really good possibilities. So thanks, Lynn. Yeah, we'll check that out.
There are fluorescent UV light systems for disinfecting water. Would these work?
Yeah, great question. Again, I don't know much about those specific systems. It's going to depend on the wavelength of the light that those produce and the intensity. It's a lot of it is about the delivering enough energy to actually get decontamination. Without knowing the details of the systems, say maybe. It depends on wavelength and energy.
And how well would UV work with homemade cloth masks, or is simply boiling them in water for a minute sufficient?
Yeah. So UV, I would imagine, would work just as well on the cloth masks as it does on the N95s. The one thing I don't know about, the cloth masks are quite a bit more permeable than the N95, and so there might be some issue with penetration of the UV into them. Boiling is easy and accessible and fast, and so boiling them or running them through the wash is fine.
And I would suspect that hospitals are not going to be using cloth masks, and
exactly.
home is not going to have an easy access to UVC. So
exactly.
I suspect the other issue with cloth masks is simply the two are not going to meet.
Exactly. Yeah, I agree.
But an interesting thought experiment.
Yes, absolutely.
And as we discussed yesterday, I've been in the habit of leaving my cloth mask in the sunlight for a few days, and I know the time is doing it. But I tend to think, the ultraviolet, breaking the RNA bonds, why not? Okay, there are open source UV disinfectant projects using off-the-shelf components that completely isolated UV from the user. Is there possibility that we can send the completed product to you or someone else to check their effectiveness? Oh, I presume someone building their own and looking for some way of verifying that it's effective.
Maybe. I think I'd need to know more about what the project, so what the product is. So I'm just staring at that question. I need to know more about what the product is to get a handle on it. I had meant to put my contact information at the end of the presentation and didn't, but I should be easily locatable on the web. So if this is something you want to follow up on, feel free to send me an email. I don't think I can give you a solid yes or no at the moment.
It would certainly have to depend on the protocol of what the person meant by effectiveness and what was being tested, because of course, the university in good conscience could not allow someone to be sending in a potentially contaminated piece of equipment onto our campus where we have people. But under a controlled testing of a surrogate, such as Alex is doing with the E. coli, all of those sorts of things would come into a question like this.
Absolutely.
And that touches on some of the reasons for Science Café as well, is people often try to contact someone at the university saying, oh, I have a great idea, I need you to test this, and are puzzled or hurt when that doesn't always pan out, although it does in a surprising number of cases. I would say probably more frequently in engineering than in science, simply by the nature of their work and their research. It's closer to applied and closer to the sort of thing that someone is likely to think of on their own. But all of the controls that need to be in place can be rather daunting.
Absolutely.
But that's not to say that public collaboration and public science doesn't happen, because it certainly happens, and it happens a great deal. And it happens more than the people who do claim that we are isolated eggheads would like to admit. So I shall get off my soapbox and move on to the next question, which says, is a HEPA vacuum filter comparable to N95 in terms of its filtering capability?
I don't know off the top of my head, to be honest. Yes, I mean, I couldn't say for sure. And the two things you'd want to look at are for, a standard HEPA, what's the particle size that they're filtering and what's the sort of percent of that particle size that are being filtered. And I don't know what the standards are for HEPA, so I'm sorry, I don't have a
I have a feeling I knew that once, but I don't remember off the top of my head either. I worked in filtering, but not in air and gas filtering very much. Right. And it was a very long time ago. I do know that people are told with cloth masks that a bit of HEPA vacuum filter put in a pocket of the cloth mask will boost its effectiveness and make it more difficult to breathe with. So there's always the toss-up, if you put solid plastic film, you will filter out particles, you will also smother, so do not do that. But I know I've heard that HEPA increases the efficacy of a cloth mask, especially if you have not been able to get the tightly woven cotton and the polyester layer that they say helps with adherence.
Yeah.
But I don't remember HEPA anymore either. I mean, there are commercially available UV floors you can test. Sorry, this isn't a question. Okay, yeah, I'm not sure. All right, so we do see, and Liam Golding, the one that I accused of putting Alex on the spot in public, also noted, sorry.
That's no problem at all, Liam.
As I said, I was not criticizing him, I was praising him. I always invite people to put our speakers on the spot, and Absolutely. When we are able to meet physically as well as intellectually again, I always point out at the end of the formal questions that the after-class scrum in academia is often a good time to get questions answered. And so I always point out that I'm going to throw my speaker under the bus at that point and invite people to mob them. So Liam, don't think I was criticizing you. And we have another question that has come in. When evaluating efficacy of bioburden, literature for UV or other methods, what do you advise when considering non-N95 decontamination? So the decontaminating of non-N95 masks, I assume, is being asked. It's Liam again.
Yes, no, absolutely. So I may or may not. I see. Okay, right. So you're not necessarily okay.
Okay. Surfaces.
Yeah, gotcha. I think there's a few things there, and maybe this is something we can talk more about it. So where do I start? So I think one thing I suspect there is that different organisms may respond quite differently. So SARS-CoV-2 might respond differently from a bacteria that forms a biofilm on that surface, for example. Biofilms are sort of mats of bacteria that adhere to a surface, and they can be quite resistant to decontamination and to antibiotics and all this sort of thing. So that's something to consider in, there may be differences between organisms on solid surfaces. Another thing that I think needs to be looked at when looking at the studies is how the organism is actually being collected from the surface. So often, people will use swabs to collect organisms from surfaces, and does that, how well validated has a particular swab been for that surface or for that organism. Again, there may be differences that are have more to do with how well a particular swabbing technique picks up an organism than the decontamination itself.
Okay, and we have run out of time and questions at about the same point. So I will once again thank Dr. Alex Wong of the Carleton University Biology Department for the presentation, and thank all of our participants for joining us for Science Café. We will be keeping you posted on the next ones. I should have pulled up the schedule because the next Science Café is already scheduled, and I did not pull that up from my records, so I can't announce it now, which is unforgivable of me. However, it gives me the opportunity of telling you to continue visiting the Office of the Dean of Science webpage and keep track of all the things we're doing, because there's plenty of interesting announcements going on as research ramps back up that are posted there in writing. And we will have more science cafes coming up in future. Those of you who've joined our various mailing lists will be notified of them. And I look forward to seeing you in virtual form at our next Science Cafe. And in the meantime, stay safe, and for goodness sake, go and wash your hands.
Thanks, everyone.
Goodbye, everyone. Thank you.